Genome-wide association studies are a way for scientists to identify genes involved in human disease. Here, we see 5 healthy individuals, but the 6th has a disease such as cancer, schizophrenia, or diabetes. We want to figure out what genetic variations might be contributing to the disease. Data from these types of studies can help to predict an individual's risk for the disease and could even lead to a way to stop it. Genome-wide association studies look for variations that occur more frequently in people with a particular disease. They begin by detecting which proteins could be differentially expressed and predicting which genes are responsible for this differential expression. For example, perhaps all 5 healthy individuals produce protein X, but the 6th person has a disease and produces an altered form of protein X that we'll refer to as X prime. This altered protein might be associated with that disease.
Genome-wide association studies can be used in a variety of contexts. Poison dart frogs present an interesting example for researchers to use genome-wide association studies to look at genome variations that contribute to toxicity in the frogs. Poison frogs do not produce their own toxins, but sequester them from the prey that they eat. Because of this, frogs raised in captivity and fed non-toxic prey are themselves non-toxic. The genes involved in the toxin sequestration pathways are likely to be differentially expressed in these non-toxic frogs as compared to wild-caught, toxic frogs. We are trying to identify the genes and proteins that are involved in this pathway, and to do so, we use a genome-wide association study. To begin our frog toxin transport studies, we compile a transcriptome that represents the total sum of all of the messenger RNAs expressed in the tissue samples. RNA is reverse transcribed into cDNA, and the cDNA fragments from individual frogs are compiled into one long reference transcriptome. The reference transcriptome is compiled from samples from both toxic and non-toxic frogs to ensure that genes that might not be expressed in one population are accounted for in the transcriptome. Once this transcriptome has been created, the cDNA sequences from each frog are compared to the transcriptome. The number of times the sequence for a gene is detected in the frog is counted and reflects the expression level in that frog. The same process is done for each frog for all genes in the transcriptome. After the alignment has been completed, we can compare the overall trends in gene expression in toxic versus non-toxic populations. Some genes are not differentially expressed, like gene B, while some are upregulated in the toxic frogs, such as genes A and D, and some are downregulated in the toxic frogs, such as gene C. Genes that are differentially expressed between the two populations are considered candidate genes that are likely involved in toxin sequestration and transport.
Once we have identified the differentially expressed genes, we can compare their sequences to a database of known protein sequences to identify candidate proteins that may be involved in toxin sequestration and transport.
Genome-wide association studies can give us a shorter list of candidates to target out of potentially thousands of genes or proteins. However, on their own, these studies are not enough to determine causality. Further functional studies need to be done to support these potential correlations. One possible functional analysis we can run for the frogs is to separate components of their blood to find the highest concentration of toxins, and then look at what proteins are found in this portion. We can subject this fraction of the blood to mass spectrometry, which is an analytical chemistry technique that identifies the amount and type of chemicals present in a sample. In our case, we could identify the frog proteins in the fraction of the blood plasma with the highest concentration of toxins. Proteins that have been identified in both the genome-wide association study and the mass spectrometry are very likely to play a role in the toxin transport process in poison frogs.
Because genome-wide association studies identify differential gene expression across the genome, they also represent a promising way to study complex human diseases such as cancer, schizophrenia, or diabetes, in which many genetic variations contribute to a person's risk. Once we identify these differences, we can better understand how they contribute to the disease phenotype. Moving forward, we could design genetic screens to assess an individual's risk for a disease and also develop potential remedies like pharmaceuticals that can restore the desired phenotype.